So we've also been able to look at the areas of the genome that you can only look at with whole genome. So we've looked at complex carrier type. Here we just pick 4 copy number alterations. It is an arbitrary cutoff. You can vary that, and we have further work being developed in order to find a more accurate cutoff. Here we find that individual patients with a higher number of copy number alterations obviously do significantly less well than those patients with fewer. And we also find a strong, significant effect of the total length of those copy number abnormalities, which actually seems to be almost stronger than a pure count. So obviously you see that patients with longer copy number abnormalities in total do less well than patients with shorter. So on top of this, there's also an analysis from 2013 by Alexandrov, which allows you to look at all the SNVs within a somatic patient sample for a tumour and use this to characterize signatures in the DNA that can be linked to prognostic effects. So signature 9 is one of a number of signatures that we think is caused by the AID enzyme. So our reasoning was perhaps we can use this as a proxy for the IgHV immunoglobulin status as done by Sanger. So we can see from our data that there is actually, out of 97 patients, there are only 8 that were characterized differently between the Sanger and the presence or absence of signature 9 in that sample. So that's really, really good. And when you look at the Kaplanmeier curves for the entire data set, you can see very, very strong separation between the mutated and unmutated patients, the same as you would see with the Sanger sequencing. We've also been able to look at telomere length, and we see that long telomeres, patients with longer telomeres do better, which is supporting previous research. So the main idea behind what we're doing at the moment is, okay, there's a lot of markers that we're coming up with. How can we group them all together into something that is diagnostically, prognostically useful and a lot simpler than having marker after marker after marker? So we use a method called non-negative matrix factorization. That was first designed for facial recognition software. So it takes the underlying data. For facial recognition software, it would be the photograph of a face, and then it breaks it up into 2 matrices. One represents the main features of the data, so eyes, nose, face. The other one represents the importance of each of those features. And then combining those matrices, if you multiply them, you're able to recreate the existing face. And the way that that's applicable to our data is that it allows you to take this matrix of information and break it down into 2 matrices that capture the underlying structure of the data. So that's what we've been trying to do. We've taken all the markers, such as specifically mutated genes of interest, IgHV, signature 9, telomere length, copy number alteration, and then we've used that information to perform non-negative matrix factorization to produce groupings that explain the underlying structure in that data. So in doing this, we've managed to come up with 4 novel prognostic groupings. The worst patients are obviously those with the TP53 mutation. I shouldn't say worst patients. The patients who do the most poorly have TP53 or 17p abnormalities. And then in the middle, there are 2 significant groups that split out. And then we've managed to find a final 4th group that do actually better than the immunoglobulin mutated patients, and that is significant. So the beauty of this technique is that because it's WGS based and it's all bioinformatics analysis, you can put it together into a pipeline so that you can produce these prognostic groups at the click of the button. So it could potentially someday be used in a surgery, in that you would have your VCF file, you would click and you could assign your patient into one of these groups. So that's what we've been doing.